Well, thank you. And thank you, Christina. And I do actually have to thank Christina because it was Christina's idea that I take on this commission and make this piece about Kandinsky. And she spoke to me, I think, about 3 years ago. I'd made a couple of films about painting for the National Gallery of Art. And she asked me if I'd be interested in making a film using this very special video playback technology that's been developed by Isaac Dimitrovsky that's in the audience to look at Kandinsky, paintings by Kandinsky. Kandinsky was not an artist, I have to say, that I had an immediate attraction to, although I look at a lot of paintings. Kandinsky was not an artist that spoke to me right away. And it just happened that there were 3 paintings. I was in London, and there were 3 paintings from the Guggenheim collection in the Tate. And I went and I looked at them, and I looked at them, and I honestly felt that I couldn't quite find my way around these 1913 abstractions. And then I thought to myself, actually, I can use that idea. Maybe I can see how other people look at this work. So I got together with my producer, Roberta Friedman, who was also in the audience, and said, do you think it would be possible for us to find a way to do eye tracking of these paintings? And Roberta helped me a lot. She found the scientists, Dr. Ciufredda and Dr. Zikos, who you'll also hear from later, and they invited us into their studio. We made a full-size print of one of the Kandinsky paintings and invited a number of different people in to look at it and tracked the way they looked at it. And that's what you'll see in this first section of this 3-part film. Now, I have to tell you, the commission was really great and really open. And they took me to their warehouse, the Guggenheim warehouse, and let me look at every painting that they had in the collection. And I found myself most interested in these paintings from 1913, which was the moment that Kandinsky was fully committing to abstraction. In 1911, he said, abstraction is only for ties and carpets. And we're very happy to see that the Guggenheim actually took him up on that idea. And Dr. Ciufredda, you'll see, is wearing a Kandinsky tie. And then in 1913, he actually, in one of the 3 paintings I looked at, committed fully to abstraction. So you will see at the 12 o'clock screening how we dealt with that. So in the piece, Kandinsky, A Close Look, I looked at 3 paintings from 1913 in very different ways. And we will see that at 12. But right now, we're just going to look at the first part of the film, which deals with eye tracking. And without further ado, let's show it.
I want to thank the Guggenheim and the Graduate Center and Grahame for putting this great forum together. I'd like to start with why do our eyes move? We have to, our eyes don't see the same way everywhere. Our vision is not as sensitive everywhere in our visual field. Our center vision is very sensitive, and even within 1 degree, we lose about 60% of our sensitivity. So we really have to move our eyes in order to be able to perceive the scene as a stable and clear environment. This is an example of how our visual sensitivity changes or varies across the field. The center, we can perceive very small letters, and as we go even a little bit outside, the letters have to become much bigger in order for us to see them. Our eyes move. Each set of eyes has 6 muscles that move it in any direction that needs to move. and they move together very well coordinated fashion. There are many different types of eye movements. There's eye movements that always occur, that they're called microsaccades. There's vergence eye movements where something is moving closer in space. Our eyes will turn together to look at it. And there's what's called saccades, which are very fast movements that are coordinated when we look from one area of the picture to another. And also there's smooth pursuits when a target moves smoothly across our visual field. There's a very large part of our brain is used for our visual perception. Not only the visual areas, but in order to be able to perceive and guide all these movements, and I'm not going to go through this, it's a very complex process. There's all the visual areas that process the picture and give us different features, but you need input from our attention, our memory, in order to be able to move our eyes appropriately to be able to perceive our environment. I'm going to run through quickly some of the history of eye movement recording. Back in the 1980s, initially the Javal had discovered and named the eye movements we call saccades during reading, and that was done with visual observation. After that, early 1900s, high-speed photography of the corneal reflection gave ability to measure more and understand how the eyes are moving, and pursuit and eye movements were studied that way. And later, especially around the 50s, there were more heroic experiments where they actually would put plaster or plastic scleral contact lenses with mirrors on top, and we're trying to understand how our eyes are moving and record the output in film. Again, in the 50s, we also had another way of looking at eye movements. Our repertoire added ways of recording muscles and the movement of the eye with electrooculography. It's still used today for studying vestibular problems. Another way, again, that started in the 50s, but is still used today, there's a couple of commercial instruments that are easily used, is infrared reflection techniques. Dr. Ciufredda uses that in the clinic in training people with brain injuries and people with different types of conditions like nystagmus. It's a very nice, simple technique. It has some limitations. For example, it only records horizontal eye movements. For great accuracy, in the 70s, we were able to use magnetic search coil contact lenses. Again, it's not the most comfortable way of going about it. This contact lens that has a coil and actually a wire is inserted in the eye, and then you you move your eyes. It has the advantage of having incredibly high resolution. And then we're going back to some of the original techniques, but now with much better tools. So instead of using high-speed photography or movies, we're using video-based systems. And this is a system that we are still using today. It just doesn't take half a room anymore. And this is the system, the company is called iScan, that we used for Grahame's project. But now, instead of having big cameras and all this computer processing. Here, everything can be done with either small cameras that can be attached on glasses or with a small computer. This is used for many types of research these days, and not just research. It's used in advertising. It's used for input for computers, and I know we'll have a lecture later on with all the applications of eye movement. And I'm going to go through a little bit how this is done. The eye is illuminated by an infrared sensor, and it's captured as a video base. And the pupil, we find the center of the pupil, and that's what the eye tracking software does. And also, there's a reflection of the lights, and here we have a single light off the cornea. As the eye moves, the center of the pupil moves, and the corneal reflection also moves. And the relationship, I'm going to go back, the relationship of these two allows us to know exactly how much the eye has turned and where the eye is looking. And I know we just spent some time, but this is part of the software that we're using. There's a camera that looks at the scene, and this is what's called the point of regard, basically where on the scene the eye is moving. And this tracks both eyes together. And once you do a fairly quick calibration, then you know exactly where the eye is looking. This is recorded, and then you get these beautiful processed images, so we can understand exactly how and where our subjects are looking. For this project, we had used a fixed camera system. It's a very simple system because all you have to do is put your head in the area that the camera is looking. But as I showed you before, this can be also by portable systems, just by using cameras on glasses. And that's my quick review.
Eye tracking doesn't get enough publicity and fame, but it's obviously really important for everything we do in life. I wanted to touch upon a little bit more some of the things that Dr. Zikos also alluded to. This first, I'm going to press the right button, here we go. This first technique, Javal was a very famous vision scientist, ophthalmologist, and prior to that time, when people read, since your perception is continuous as you read, people thought that you made smooth eye movements, that your eye moves very smoothly as you read, because your perception when you read is not that you're seeing isolated words, but that you're seeing a continuum. And he did an elegant experiment, shows that you don't need $2 million in NIH grants to really get insight into how we do things. And he simply observed, put a mirror below the text that someone was reading, reflected their image of their eyes in the mirror, and over their shoulders would look at the eyes. And he, lo and behold, he found that you didn't make smooth pursuit movements, even though perception is smooth. You make a series of saccades, jerks of the eye, to look at the areas of interest, and as George said, to move the fovea, the high-resolution part of the eye, to see fine detail of whatever image you're looking for. So it was an insightful experiment that cost probably in 1879 like 3 cents for the mirror or something like that. So the best computer in the world is not the one we have here, but the one we have here in our heads. These aerobic experiments are kind of interesting. These scleral contact lenses go back to the late 40s, early 50s, where you put a huge, not like your little 10, 12 millimeter contact lens now, but a large one on your eye, made of plastic, lucite. And it had a stalk coming out of it, about a ½ inch or so, and a mirror on it. That mirror reflected onto some photosensitive materials. So it wasn't exactly the most natural way to look at things, but it certainly gave you high resolution. But you had this thing on your back, on your eye, while you lay on your back watching how they measured fixation. How do we fixate and how accurately do we fixate? Oh, sorry. And as George said, this is another technique, EOG, where the eye is a dipole. So the front of the eye is positive relative to the back of the eye. So as you move your eye, these little electrodes will pick up electrical signals correlated with horizontal and or vertical eye position, depending on how the electrodes are pasted on in the appropriate manner. It's a simple technique. It's not a high-resolution technique. Well, this is a system. Stark was my mentor at Berkeley about 30 years ago, or so, 35 years ago. And he, after World War II, there was this revolution or evolution in terms of cybernetics. Norbert Wiener, McCulloch, Pitts, and others, and Larry was part of that scene, or later part of that scene, where he developed the eye movement system, not to develop an eye movement system per se, although he did make it a commercial system later on, but to really investigate how we track objects, artwork as well as other things, and convey it and describe it in mathematical terminology. So equations, Bode plot analysis, those kinds of things. And as George said, what this is doing is looking at the infrared reflected light off your eye in the 3 and 9 o'clock position. And as your eye moves, the amount of reflected light from the, say, brown-white region temporally versus the brown-white region nasally would be different over a range of 10 or 15 degrees. Yeah, this is another heroic technique. We've used it a little bit in Sweden, and it makes people cry, that's for certain. And after about 20 minutes, some people have pain because it's an angular, thick, soft contact lens with this wire coming out. And it also gives people a lot of pain, and the intraocular pressure of the eye seems to increase because it's pressing on and sort of sucking on the external brown-white region, which presses on where the aqueous fluid of the eye is eliminated. So it's not good for people with glaucoma, that's for certain. And as George said, this last one, this is sort of what happened in the 80s. All these cameras can now be mounted on spectacles, and people can mount them on spectacles and use them for advertising purposes, walk down an aisle. Where are people looking? Are they looking where the signs are? Are they looking with the new product, the way the packaging is done, et cetera? So it has commercial applications, which are important, as well as just scientific applications of how we perceive the world. And I'm going to skip what George went over. The person in the picture here, film here, is my daughter, who's also an optometrist. And briefly, she got me the job. Roberta went to see her as an optometrist got a vision checkup, and then Roberta asked my daughter, gee, we're going to do an eye movement project. You know anyone who knows something about eye movements? Says, yeah, my father. So that's how I got the job. So I have to thank my daughter at this forum. Second part of my talk, or sort of carryover, is just what I said. How do we perceive art? Not only how do we perceive art, but how do we perceive the world that we live in? And there's sort of two hypotheses that have been put forth, and we'll discuss each one, although I think from what I said at the end of the movie, that art drives the visual processes and drives the oculomotor system, you'll know which hypothesis I support, even though the other hypothesis is by my mentor. Unfortunately, he's not here to argue with me about it, but if he were, he would argue with me, and who knows who would win? Probably be a tie. So the first approach is the bottom-up approach. And this is, those of you who are psychologists and looked at infant perception and things like this in the 40s and 50s and 60s, people were interested in sort of regions of interest. If I put a triangle up, where does the baby look? And the baby doesn't look at the lines, looks at the apices of the triangle, looks at where the action is, where's the high information content, regions of interest. They're not going to look at the center of the triangular form or square or something like that. And one person who was an advocate of this was Jarbis, who was a Russian physiologist in the 60s and 70s. And he, basically, his idea, and these others, says that the external world, or the stimulus, drives the oculomotor system, and we have regions of interest, high information content, that we look at. So let's look at some pictures. This is a picture called the Volga girl. from Yarbis's book, Eye Movements and Visual Perception, and he had people look at this picture for more extended periods of time, not a few seconds, like we'll show in a minute, but probably a minute or two, a little bit like we used in the film. So the question is, where do you think a person looks when they look at this girl for a minute or two? That's where they look. Sort of Halloween movie, scary movie or whatever. But basically, the regions of interest. The eyes, the mouth, the nose, the part of the ear, and a little bit more on this side than this side. This side sort of illuminated to a little bit too much relative to here. So there's more detail that is visible here. Here it's sort of smooth out in terms of texture and detail. Another picture. This is interesting. Where do people look here? I was wrong, that's for certain. So those circles are where most of the time the individuals, the subjects would be looking. And then what was interesting here, I guess my biology background, I started be looking here. He's slicing the arm open, and I'd be looking at the person, hopefully, or maybe it's an autopsy, I'm assuming, but or highly drugged up with alcohol at the time. But people looked at the various faces. Faces are really attractive. Infants, they like to look at faces, 2-3 months old. As soon as they develop binocular vision, stereopsis, vergence, eye movements, etc., they begin to really look at things quite intently, especially at faces. And this is sort of a color imaging technique to show where people are looking. Red means they look there the most, blue means they look there the least. And it agrees with what we just saw. Just another way to visualize what's happening. This is an interesting picture. One might guess where you might look in this picture, at least where I might look. But anyway, let's see where people did look. Sort of where I might look, I would think, okay? It's interesting, not too much gazing around at the scene. But if we take a broader view where they do more fixations and use that color coding technique to show where the action is. Okay, here we go. You can see that they do look primarily here, but they do look at some of the outer aspects, especially the person or whatever in the background. So it's a nice way to to visualize what's happening. Now, so that would be the bottom-up approach. Larry Stark, always to be a little different in his point of view, really an ingenious guy. There's a picture of him in his later years. Had a different idea. They published 2 papers, one in Scientific American, sort of the easy-to-read version, and then in Science in 1971 by Milton and Stark. Milton was his PhD student. And they took a different approach. They said it's a top-down approach, that we have for various objects or categories of objects, let's say face, car, hand, etc. I have an internal cognitive model, a sequence of fixations that I've made by my visual experiences. I put those in my somewhere in my brain. And then when I see something, I then make the eye movements to confirm my internal cognitive model of what I think I'm seeing. So it's sort of, in a way, a reverse way to look at it, top down. So I make the sequence of eye movements in a certain sequence and in certain pieces or parts of the field that would just tell me, yes, the thing I'm seeing, face is really a face. And that was Larry's notion. And I'll be done in a second. So this is one of the pictures you'll probably see in Scientific American or some Nature or whatever. Those who are psychologists interested in visual perception, this picture and a few other ones are in many, many textbooks. People either like this hypothesis, okay, I'll be done in a second, or hate this hypothesis. With Larry, that was Larry. Either you really loved him or you went after him. So it was kind of vicious at times. But anyway, what we see here is that this is the scene, a simple scene, and this shows depictions of scan paths, these sequences of featuring hypothesis, sequences of movements that you have to make in a particular order on particular features of the thing you think you're seeing. in order to confirm his hypothesis. And this was some of the time the people here were the same person, was looking at the picture, or they were recalling a remembrance or memory, short-term memory of the actual picture. Scan paths are pretty similar. And if we looked at the sequences, that would be more or less appropriate. So it sort of suggests that he might be correct on it, although it's a really controversial thing. Still, people disagree, or they love it a lot. This is another one of the images that they used. These are totem scan paths. So these are 3 images, and this is how subject number 1 looked at these images, and this is how subject 2 looked at these images. So for the scan path idea to be correct, not only would the same person have to use the same scan path or internal cognitive model to perceive the external world, but they have to do it over and over again for the same person, but also the same thing would have to be true for different people. And Larry would argue that this is pretty strong evidence these 2 people are making scan paths that have a fair degree of similarity, not 100%, but good enough for Larry to think it supports the scan path notion. I don't think people have done the right experiments to really put forth and substantiate the scan path idea, so I would sort of go with the former idea, that is, the art drives perception and hence drives our eye movements. Now, there have been some compromise positions where some people say, well, when I look at a piece of art or a scene like this, I might, or let's say a piece of art, I might look at a face in there or an animal or whatever, and for that recognition, I might make my little scan path. But then for looking at more details of the scene, as you saw in the artwork, in the film that Grahame produced, that people might look at other regions to get details of it, but to get the particular image, an eye, a hand, a face, a car, an animal, they might use a scan path. So my conclusion is the debate goes on, although my position on it. I wish Larry were here. He'd be in the back there waving and giving me a really hard time, but I know that wherever he is, he's doing the same kind of thing. So I'd like to thank you very much.
That was really great, and I'd like you all to notice Dr. Ciufredda's tie, which is, of course, a Kandinsky abstraction. I guess I'm just going to ask you guys one question, and then we're going to open up the audience, and it's in relation to the film. What do you think we accomplished in terms of understanding both the art and the science in the film, if anything?
Well, I think that the interest in the film was the various characters, the various types of people that view the art, starting with the naive observer who was an artist himself, but naive as to Kandinsky paintings and probably naive to a lot of abstract art, I'm guessing. And yet the scan paths that he made, or fixation patterns he made, everyone sort of made kind of similar patterns. Even the last woman who was an expert on Kandinsky, she was talking and saying, well, I'm looking here and I'm looking here.
Yeah, that was Tracy Bashcroft.
Most of the time she was still, everyone went back to the little hill with the people on it, or whatever you visualize those things to be. Everyone. So I think it does support the notion of the art driving visual perception and hence driving our oculomotor system or eye movement system, not vice versa.
What about from the point of view?
Attention also comes into it. One of the subjects we used, which was a restorer, looked at the picture in a different way because was looking for a different type of information from the picture. So our attention it drives the system, and certainly art and abstract art has a way, because there is no internal model. You're looking at it. It's not representational art. It has to drive your attention and your oculomotor system around it to give you the emotional and the interest areas.
And the curator, the art restorer is really interested. Everyone sort of looked around the painting here and there, and this young lady went to the corner.
And look,
So here's the
at the
painting,
edges
and
and
she's at the corner, and she's looking at the
I'm afraid
corner.
I cut her out.
And looking at the, yes, and looked at the corner and looked at the corner. So a very atypical thing, because I guess she was looking to see whether it needed restoration, or maybe she had done some restoration there. Whatever, she was totally different, and just a different mindset in terms of what she was seeing, or what she thought was important.
Good. So questions from the audience? Bob?
I'm just curious if there's a way of looking at a painting that may not be able to be recorded by your technology. In other words, one of the ways when you're looking at a Kandinsky, there's an overall gestalt composition, and so I know that for me, part of the way, some of the time, some probably very small percentage of the time that I'm looking at the Kandinsky, I may sort of defocus to try to get this sense. And I wonder if that would record as simply concentrating my vision on the little hill structure.
Well, I think that if you did that, it would just show you're looking someplace, sort of spacing out, or whatever term you want to use, to get a globality, a global nature of what the painting's like. But I think it was so consistent amongst the people with different kinds of backgrounds, always being driven to the hill. And according to Kandinsky, most of his paintings had two, but sometimes one focal point. So in a few of the paintings, there were sort of two areas here, and people would look back and forth between those two. So I think we wouldn't, I can't answer that question exactly, yeah.
Focus, then you're really looking at the hill, but you're looking at sort of everything.
Well, you might be thinking you are, yeah.
Well, that's an interesting.
Where your attention is, so I can look at you,
Yeah.
my fovea, George talked about the fovea, and I could pay attention to the woman in the front row. So my fovea is here, and usually my fovea, and people use the term attentional spotlight. Imagine I have a laser pointer, but not really. That's just a flashlight. So my fovea and my attention would be in the same place. When we read, when I look at a word, my fovea and my flashlight are in the same position. But when I think about going to the next word, my fovea is still here. My attentional flashlight has moved over. So where your attention is could be anywhere relative to where your fovea is. But you have to look at several subjects, and not just one, as we did.
So you can't measure that.
You can't, unless maybe whenever the person said I want to look at the global thing, I'm going to space out, or whatever it feels like. I want to get the overall impression and don't want to look at the hill, and I don't want to look at the little thing on the right and look at the little banana, sort of. You could have press a button saying, this is what I'm sort of just gazing at it, trying to enjoy it globally rather than locally. You could do something like that.
But you see, there are, in a way, two answers to that question. I mean, I thought about this a lot. First of all, in one of the visualizations, what size am I going to make the area that we say is the notion of focus? And if somebody stays in one place for a while, do I open up, perhaps? Do I zoom out? Also, of course, we had people fixed, so they couldn't move in relation to the picture. So in a way, this was a very limited, from my point of view, a very limited study of the way people actually look at the picture. But it did go with Kandinsky's idea of kind of taking a walk through the picture. So yeah, I mean, I don't think we ever pretended that it really gave you the entire experience, but it did invite future viewers to consider the way they were looking at it and maybe kind of disagree with the way we visualized it. I think your point is very well taken. Yes.
Could you use the microphone, please? Thank you. It seems to me that Yarbis also had done a series of analyses using a Russian painting, a very famous Russian painting, where he asked the people looking at it different narrative facts about it. Wouldn't that have answered or gone towards answering the question of bottom up or top down?
I guess it could, but one of the problems with the Yarbis, he did great work, and I love all of his work. I appreciate that you're familiar with it and know a lot about his work. But just like in the Volga girl picture, the fixations were there for probably 2 minutes or 1 minute. So there might be 50 or 60 fixations looking at it. And the scan path idea would be, where do I make my first 7 or 8 or 10 fixations? Because if I have an internal cognitive model of face, then I don't need to get every detail, every hair on your eyebrow, just need face. So you really need to have a 10-second or less viewing of the image to really separate the two out. Otherwise, you have too many fixations, and you don't know this, and the person might repeat and repeat and repeat themselves. And if you looked at the sequences, you might be able to get to it. But if you have too many fixations, it's really hard to.
And who had done the fixations and measurements of the man-aid?
I don't remember, quite frankly. I just don't know.
We can only have one more question, I'm afraid to say. We're really rushed, so.
Hello. It's really fascinating work. Thank you very much. As a physician, I'm interested actually in the application. I'll make a comment and have a question about using this technology and having portrait painters who regard painters and the subject and transferring this information back and forth and trading radiologists to analyze a radiograph in a very methodical way.
That's a great point. In fact, Larry Stark, one of his PhD students, was a dentist who was interested in how do we look at dental x-rays and things like that, and use the scan path idea and just fixations. So that would be an interesting idea to sort of get them, train them to of where they should look, and not just randomly say, look at the x-ray of the chest or whatever it might be, and just look at it and look at it. And of course the resolution on a lot of those is not so good, so you'll be looking more globally at bigger kinds of things than a millimeter object. But I think training them to do so would be really
Feedback
great.
is a great tool for training. And
Thank you. And the question is,
it's something.
I'm a physician as well as a contemporary arts curator, and wondering about bias in these studies. If you're doing data analysis, where, I mean, one comment was that the Korean gentleman was not totally naive. You labeled him naive, but he was also labeled as a film director. So
Right.
that naivete is really not.
Well, he wasn't familiar with, he never saw
Right.
Kandinsky before.
So
So
the basic
naive
que-
with respect
yeah.
to Kandinsky per se, and I think naive with respect to a lot of abstract art, but that part I'm guessing on.
Sure, okay. But getting back to data analysis, what are you considering your control group then?
Well, I'm going to say this wasn't an experiment because we didn't have enough subjects. This is sort of an art movie, a documentary about Kandinsky, giving you some
Right.
insight. So we didn't have 30 subjects or 50 subjects and things like that. And we didn't limit their viewing time to, 50 seconds only or 30 seconds or 20. So I wouldn't call it a formal experiment on the topic. So there was no control group per se. I'm not certain what a control group would be like.
Yeah,
in
that's
this
my
case.
question.
Yeah,
If you're
what
doing
would the
data
control group
analysis
be?
on this,
What would a control group be in that case?
I have a little comment about that. It was true that Dr. Ciufredda, we would ask each of the subjects questions afterwards, and I would just ask them whatever questions I felt like asking. Dr. Ciufredda asked each of them exactly the same question, kind of pushing it. It was very interesting for me to work with a scientist because he was kind of always pushing it into the scientific realm, and I was kind of pushing it into the expressive realm. And I don't know if that tension quite comes through in the film, but I think it is one of the things that animates this project and makes it so very interesting.
It's fascinating. Thank you.
Thank you.
I think we have to move on, right? Thank you very much, you guys.